And record. Right. Hello, everyone. Welcome to session 9 of this year's BGA. This session is on Genespace by John Lovell. Please take it away, John.
Thank you. All right, thanks all for coming today. I'm pretty excited to talk a bit about, my software Genespace and kind of comparative genomics in general. I think maybe before we get into actually running the Genespace R package and producing our own results, I wanted to just give a quick background on why we have Genespace and why comparative genomics is, a really exciting field now more than ever. All right. So, first of all, a quick introduction. I'm John Lovell. I am a research faculty at Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. And I also am an affiliate scientist at the Department of Energy Joint Genome Institute in Berkeley, California. I work remotely and live in Denver, Colorado, in the US. And my focus is primarily on, and my group's focus in general is primarily on figuring out how to make multiple reference genomes and genomes in new species or new systems directly usable to our stakeholders. That might be breeders or people doing trait discovery or understanding dynamics of pathogens or ecology and evolutionary questions. And what that boils down to in a lot of ways is the integration between comparative genomics, comparing two genomes, and population or quantitative genetics. And GeneSpace is one way to do some of these integrations. So, as I think probably many people who are watching this here uh, are aware and were influenced by this, the original Human Genome Project from the 1990s and early 2000s was a really foundational scientific enterprise and produced what we called either the draft or the finished human genome early in the 2000s, and that led to thousands and thousands of discoveries. Probably the single most important discovery and resource in at least the last several decades for both disease exploration and drug discovery and understanding human evolutionary biology. Now, that project took, what, decades, thousands of researchers, hundreds of millions of dollars or more to do. And, it was incredibly important, but also important to know that recently it's become possible to build uh, even more complete reference genomes with substantially less cost. 2 years ago, the T to T human genome was produced, and that just used a haploid, essentially a doubled haploid cell line. So it was a much simpler biology question of assembling that genome, but also a very, very complete genome assembly of all 22 autosomes and eventually both the X and Y. This is exciting to me and to a lot of us, not just because they filled in these gaps that are highly repetitive. Honestly, the genome itself, while a huge advance, is not itself that much more important for trait discovery than the HG38 previous version. Most of the genes were already there, and most of the functional variants too. Some, of course, were not. The big important thing here is that you're able to build a very complete genome very quickly and with low cost using some pretty clever bioinformatics and new resources, including Nanopore and particularly PacBio HiFi. What this means for us is that our genomes can accurately represent biology and we can build very complete genomes, whereas we couldn't before. Here's an example from switchgrass. Switchgrass is a tetraploid outbred grass that's used for biofuels. The version 4 genome that I was working on when I first started back in 2017, that had about 2 to 4,000 contigs, contiguous pieces of sequence in each chromosome. And that was built with 454 and a genetic map and all of that. With CLR, the old PacBio, we were able to get that genome down to several contigs, maybe 20 or 30 contigs per chromosome. And now, just with the addition of PacBio Hi-Fi, we're able to build both biologically representative haplotypes. So these are the two meiotic homologs, HAP1 and HAP2, in this one genotype, and we're representing those in one or two or maybe three contigs per chromosome. So there's just handful of gaps in really repetitive regions. Essentially, this is a complete representation of the biological genome, both haplotypes. So we have not just a complete genome, but a genome that represents the true biology. Remember, like the previous human genome, HG38, that was many outbred individuals that went into one single haploid representation. And the T to T CHM13, that was built on a doubled haploid genome. So a haploid representation is just fine there. But this is what most genomes should look like, right? Two biological haplotypes. This is both exciting, but also problematic. Now we have two haplotypes, and our single reference alignments and analysis platforms are no longer valid if we want to use both. Now, the state of the art still is to build two haplotypes, but only present one. I think that that's generally a problem, and we should be trying to do everything in the multiple reference genome framework, even though it's hard. The other thing that this new technology and bioinformatic methods have let us do is build much more accurate representations of more complex genomes. So one example comes from sugarcane. Sugarcane is probably the most complex genome out there. It's not the largest, but it probably is the most complex. We know that the modern sugarcane varieties are a hybrid between Saccharum officinarum and Saccharum spontaneum. And back about 6 years ago, we built a single copy representative of this genome that has a little bit of spontaneum in it, a little bit of officinarum, and represents all of the genes in single copy. However, we know that that is not what the sugarcane genome actually looks like. It looks more like this. It has between 11 and 14 copies of every chromosome. Some chromosomes are exactly identical to each other because of inbreeding, and some are very divergent because they come from different species. There's recombination, there's aneuploidy, and there's between subgenome recombination. And this was published this year by my colleague, Adam Healy. And basically what we were able to do was build a genome assembly that represented the biological complexity of this one sugarcane genotype. In this case, this is R570 here. This is what about 1/2 of the genome should look like. And this is our previous genome assembly with shorter and less accurate reads. So it's really a tremendous improvement in our quality of our genomes. But now, where we could map everything previously to just a single representative, now we're mapping to between 6 and 12 copies of things. And that also is a problem bioinformatically and from an interpretation perspective that requires some integration. Basically, what these two examples from sugarcane and switchgrass indicate is that we're going to be doing everything moving forward in a pan-genome framework using multiple reference genomes. And we can do this, and I'll show just a quick example. Like from sorghum here, this is a project where we have, in collaboration with Jeff and Nadia, where we've sequenced the diversity across the diversity of sorghum. And if we build and look at the synteny map of these 32 reference genomes, we can see that there's a tremendous amount of structural variation and presence-absence variation. And we need to be able to connect these genomes in some reasonable way. To date, the most used method to connect any two genomes to each other are using single copy orthologs or one-to-one orthologs. Orthologs are the genes among a group of species or genomes, whatever. We don't need to distinguish species and genomes, just the genes across a group of genomes that arose in a phylogenetic context with those genomes. So for example, in the case of human, mouse, and chicken, what we can say is that, human, mouse, chimpanzee, and chicken, if we just compare human and chimpanzee 85% of all of the genes in there arose at approximately the split between human and chimpanzee, and then have been retained as single copy over the last 6 million years. Human and mouse, it's something similar. Even though it's almost 100 million years of divergence time, there's 82% of all the genes are one-to-one orthologs, meaning there hasn't been any copy number variation or gene loss over that time. And pretty astoundingly, over 2/3 of all genes are one-to-one orthologs between humans and chickens. Even though that's what, 300 plus million years of evolution. So what we can do is, given that we know that everything's basically single copy, you can easily just track regions of interest across these reference genomes using the retained synteny and knowing that you expect one-to-one relationships. So like, for example, here on the X chromosome, the X chromosome in human is 100% conserved across placental mammals. It is partially conserved in marsupials, and then is conserved but not related to sex chromosomes in monotremes or birds, but is partially related to some sex chromosomes in the sand lizard genome, for example. Same thing could be done with the avian Z chromosome. So basically, if we assume everything is single copy, one-to-one orthologs, comparative genomics is easy, it's trivial. And in fact, that's the case in many vertebrate lineages, but not so in plants. So this is what 30 million years of grass evolution looks like. I can't flip back to that. Well, I can here. Just go back to this. This is what 300 million years of vertebrate evolution looks like between birds, other reptiles, and mammals. And if we go to, this is just 1/10 of that evolutionary time in grasses. So switchgrass that I talked about earlier is up here, maize, wheat, etc. And this synteny map is obviously far, far more complex than even 300 million years in vertebrates. And the reason is because plants and many other lineages in the evolutionary tree, but not those vertebrates I showed before, plants have tremendous number of whole genome duplication events and polyploidy. So the genomes that we're looking at here, about half of them are polyploid. Wheat is a hexaploid. Switchgrass, as I mentioned before, is a tetraploid. Maize is a fairly ancient tetraploid. And all of these genomes share a whole genome duplication event that preceded the diversification of grasses. So tracking regions of interest here using one-to-one orthologs is not only difficult, but not particularly informed, in part because these whole genome duplications yield, non-one-to-one relatedness. So for example, just any two maize genomes that you might compare have about 2/3 of their genome are one-to-one orthologs. So if we think about that, just any two corn plants that you pull out and you sequence will have fewer one-to-one orthologs to each other than the 300 million years of evolution between humans and chickens. So if we're using a model that's designed for studying evolution in, for example, primates, where we're using one-to-one orthologs, that isn't even appropriate within maize. It's a little better in systems that don't have an ancient whole genome duplication, like sorghum. Sorghum, 82% of the genes are one-to-one orthologs, but also there was a really recent severe bottleneck in the diversity of cultivated sorghum. And that 82% reflects as about the same number of one-to-one orthologs that we would expect between humans and mice, approximately 100 million years of evolution. So long story short, we shouldn't be thinking about one-to-one orthologs as the way that we compare genomes. We should be embracing copy number variation, where one genome might have no genes and another genome has multiple, or just variation where there's presence of the genes in all the species, but the number of them vary between whatever, 1, 2, 5, 10. The reason I mentioned before that we have this issue with with copy number variation in non-one-to-one orthologs in plants is because they're whole genome duplication. So this is just a dot plot showing orthologous regions between two maize varieties. And you can see there's a strong diagonal. Those are the meiotic homologs. A lot of them are one-to-one orthologs, but then these off diagonals, those are the homologs that don't necessarily recombine or don't recombine during meiosis, but do have enough similarity that they pollute our estimates of orthology. This isn't just in maize. If we try and extend our data in plants from any species back to the genetic model, for example, Arabidopsis, these are two plants that are pretty closely related to Arabidopsis, Camelina on the left and Brassica rapa on the right, that have both undergone whole genome triplication events. You can see how strongly these homologs related to whole genome duplication events are present in these dot plots. Off diagonal is everywhere. And if we try and take, for example, common bean and ask what genes are related to common bean and the model plant Arabidopsis, you can see that not only is synteny broken down over 50 million years, but also there are 4 to 6 copies of every gene as you look across the genome relative to the genetic model. This seems impossibly complex. It is complex, but not impossibly so. And the way that we can make it not impossibly so complex is by focusing on regions of interest and contextualizing our expectations for copy number by the expected whole genome duplication event. So here, this is just a synteny map tracking 2 positions, one on chromosome 3 and one on chromosome 5 of Arabidopsis across 5 genomes. And we know that there's been a whole genome triplication event between Camelina and Arabidopsis. And so we see that these 2 regions here map to 2 regions, each map to 3 regions here. And then as we go deeper and deeper in evolutionary time, there's more and more whole genome duplication events, but we can still, even with soybean, who has 3 nested whole genome duplication events between it and Arabidopsis, even in that case, we can still track single positions all the way across back to a region of interest in Arabidopsis. Okay, so we can do this also in, as I showed earlier, this is a figure from the Genespace paper on larger regions in much deeper evolutionary time in vertebrates. Okay, so that is the lowdown of how Genespace should be able to contextualize complex comparative genomic situations, not just in simple systems, but in more complex ones. So maybe I just give a second if anyone has any questions here. Please feel free to ask about that little presentation, and then we'll move over to the demonstration part of this.
I just have a question. Hi,
Yeah.
John. You showed in some of those plots the synteny of some of those chromosomes, but it was just a small piece of the chromosome that actually had a duplication. I was wondering if you could just kind of comment on, I feel like the previous assumption was, no, the whole chromosome is duplicated, but the synteny plots were showing just a small piece of that. So can you
That
talk
was just because I was tracking one little piece of the genome in Arabidopsis across the multiple reference genomes. So yeah, if you look at the synteny map between common bean and Arabidopsis, that dot plot I showed with 4 mappings, top to bottom, that is what you would expect anywhere on any part of the genome. You get 4 copies, but the genomes have been shuffled up enough through structural variation across 50 million years of evolutionary time or more that it's not a whole chromosome mapping 3 times to another. Does that answer your question?
Yeah, I was just curious on, whether you're always expecting this whole piece is that whole piece or vice versa, but I assume it's always pretty fragmented. So,
well, when you compare any 2 genomes to each other and you look at any single piece and there's been a whole genome duplication, then yeah, you would expect the bifurcating tree there. But when you have 5 genomes that all have different whole genome duplication events and different structural variants, you would expect them to get chopped up like that. Synteny, the relative retention of synteny of gene order across a chromosome is really variable between species, between clades. So in birds, it's basically 100% synthetic. And in, if you look at the Brassicaceae, which contains the rape seeds and a bunch of oil crops, there's basically a complete breakdown of synteny. And that just has to do, I'm sure, with the meiotic machinery in those systems, but I don't know exactly the reason for it.
Thanks.
Yeah, no problem. All right, so we'll go over now and do the hopefully you're seeing this, the tutorial part of this session. So I guess maybe I'll give people a second to navigate to their Gitpod. If you're not using Gitpod and you're doing this on your own, where I'm starting right now is in a directory, and we can just do that. This is a directory we've already made that contains the input data that we need. Come on. So if we look at this directory that we're in, we have the peptide embed subdirectories. These contain the Genespace input files they look like this. They're just a peptide FASTA file and a bed file named genome.bed and genome name.fasta. There is a auxiliary function with Genespace that I'll demonstrate later that makes these files for you from raw GFFs and raw translated CDS peptides. If we just look quickly at these files, we can do this more. I'll just look at the chicken one. So these are very simple files. They contain the chromosome name, the start and end position, and then the simplified gene name. Same goes for the FASTA files. They actually are exactly matching. The name is exactly matching the header in the FASTA, and then it has the peptide sequence there. All right, so this is where you should be starting your run from. And yeah, if you need help getting to this position and making these input files for your own data, that's kind of one of the trickier parts, and there's lots of documentation. I guess I'll just point to that documentation right now, that this is the README. If you go to the GitHub page, and there is a lot of information about how to install the package, and then information about the input files. And there is somewhere in here, that now this is information about how to make your files. There's a function called parse annotation that can do this for you. Okay, so let's go back to our session. Okay, so the first thing that we do to get Genespace going, and by the way, people hop on in and let me know if there's an issue while you're getting this going. And I can stop and make sure that we're troubleshooting everything. All right, so Genespace is an R package, and eventually it'll be just a direct command line tool, but right now I don't have the bandwidth to write that, so we have to do it interactively in R. There's also scripts out there to run it non-interactively using an R script. So you enter R just by typing R, you get something that looks like this. Hopefully you're using R 4.4, but 4.3 or earlier is no problem. And then we load the Genespace package, which is already loaded into this Gitpod directory, but you can install it from GitHub yourself, and there's information about how to do that. All right, so to run Genespace, you need to have a valid orthofinder path, a valid path to MC scanx, and a working directory that contains those input files. You can always just check to see if orthofinder is installed using R. You have to type in system orthofinder -help. And that calls or the OrthoFinder help page, and you can see it's installed and is in the path here. So if it's not in the path, you have to give an actual path to OrthoFinder, and you can specify that in init Genespace. The same goes for MC-ScanX. So the path for that is just in the bin here. All right, and so we give it the working directory, our path to MC-Scan. Goodness. What happened here? Maybe there's an issue with the copying from, yeah, heads up when you're copying from the PDF, you need to do it one line at a time. That's obnoxious. All right, so then we start our run just with this function init Genespace. And what that does is it checks if your directory structure is all right. So we're assuming both of our genomes have single ploidy. I didn't specify that, but you certainly could. But default is 1, and it gives a bunch of parameters that we've passed to it. Here we're using 4 cores because that's what is available on this Gitpod instance. And then it goes through and checks your annotation files, and it says everything looks good. All of the genes match exactly between the bed and the FASTA file for both chicken and human. And we have valid paths to MC-ScanX, Diamond, and OrthoFinder. All right, and then to run Genespace, all you have to do is run the command run Genespace. So what Genespace does is it checks to see if OrthoFinder has been run beforehand, and if so, it doesn't try and run it again. But in this case, since I'm using a new instance here, OrthoFinder has not been run yet, so it has to run it itself. This scales pretty much exponentially with the number of genomes. With the 2 genomes here, it has to run 4 blasts, chicken to chicken, human to human, chicken to human, and human to chicken. And that takes a little while, probably about, I don't know, 5 or 6 minutes. And then it'll parse those results, and we'll go through it as we move forward. But let's go back and take this time now and talk about our input files, if we can. All right. So when you're building, if you want to build a custom input files, all you have to do is use your whatever data manipulation workflow you would use to produce files that match that format that I was describing earlier. We can also, and I'll use this time to show how we can use this function parse annotations. So if we have a raw genome, one of the main repositories that we use is Phytosome. That's for plants. You can just say parse annotations. You give the path to the genomes. It grabs anything with the GFF string and the FASTA string and the file name, and then it has a preset to parse Phytosome. Same goes for NCBI, which I guess I didn't show here, but you can just use preset equals NCBI, and then there's a whole bunch of other parameters that you could use to nail down the correct parsing. All right. I'll use this time to also talk about the other resources that are available to help you with Genespace. So the Genespace article is here. Let's just click on that and keep that there. So Genespace is published in eLife a few years back, and it goes through a lot of the details that I'll present today. Probably the most important one is just a quick overview of why we have Genespace, and I'll show you why in just going over figure 1. So this is an overview of what Genespace does. But essentially, right here on the left here, this is what the output from just feeding the BLAST results to MC-Scan looks like for a particularly complex region on the chimpanzee chromosome 6 and human chromosome 6. So MC-ScanX is really good at culling hits to synteny in places that are single copy. You get these nice synteny maps. But once you get into places where there's multiple copies of things, this is a repeat array here, for example, MC-ScanX thinks that these are all overlapping blocks of each other, all these little syntenic pieces. And that's not really what it is. What it is if you look at this, it's a big inversion, another translocation, and then a duplication in this interval. And you can see that if you just sort of squint at these spots. So if we just subset the blast hits to those that are only in the same orthogroups, an orthogroup is, in this case, with just two genomes, just orthologs, genes that arose from a single copy in the common ancestor. And if we just use MC-ScanX on that, the picture becomes much clearer, right? These things are all paralogs, not orthologs. And so we get this, but we get these duplicate regions that are not real because it's hard to know what's a paralog and what's an ortholog. And Genespace gets around this primarily by condensing syntenic arrays to a single representative of each orthogroup and then moving forward. And so this is what this region looks like in Genespace. So if you're just feeding blast hits to MC-ScanX, you're going to get tons of this. And if you use Genespace, which is just a parser for those two functions, you get a much cleaner result. Genespace then takes these data and essentially reorganizes them into what we call a pangenome annotation or a pangene set. And here's the HLA region, this complex region on chromosome 6, and it basically decomposes these into situations where you have single copy genes. This is a 2-copy gene. This is one with copy number variation across these 3 genomes. And importantly, it also determines the expected syntenic position of genes that don't exist in one or more genomes. And so here, for example, this RXRB gene doesn't have an ortholog in chicken, and yet we can predict the position that it should be in these pangene sets. All right, let's see how our OrthoFinder run is going. Oh, it's done. Sweet. So now we can talk about what the output is. So this is pretty fast. So this is what OrthoFinder prints when it's done, right? This part here. So this is just what, if you ran OrthoFinder yourself, this is what it would print as it's running through. So what it does is it makes diamond databases, does diamond-to-diamond searches. Diamond is like a BLAST. It's a BLAST-based search, BLAST similar search, but much, much, much faster, not that much less sensitive. Then it takes those BLAST hits, connects putative homologs, so anything that's similar, makes a graph out of that, and then takes those sets of homologs and splits them, first using OrthoMCL, which is what you see here, and then for any multiple copy orthogroup that comes out of here, it then builds trees and splits those based on orthologs. And so that's what it's doing here. This first part here, this is what the original 2015 or 2014 OrthoFinder approach called orthologs. No, I guess it was 2017. And then since 2019, I believe, those raw orthologs saved as the ortholog.txt file, those are now deprecated, and you should only be using the phylogenetically hierarchical orthogroups, which are the orthogroups analyzed within a phylogenetic framework and split according to rules that are outlined in these papers. All right, so Genespace then takes the OrthoFinder results and integrates them. That's the main thing that Genespace does. So it takes these orthogroups. In this case, it takes both the deprecated orthogroups and the phylogenetically hierarchical orthogroups. We call these OGs and HOGs. And you can set Genespace to use the original defunct orthogroups if you'd like by saying use hog equals false in the initial specification of the run. But in this case, OG here is hogs, not orthogroups, because we're saying use hogs equals true, which is the default. All right, so it checks all of the chromosomes first, all of the genomes first, and it says, hey, is there any problems? It's worth knowing that for chicken, about almost 500 genes are on chromosomes smaller than 10 orthogroups. And that's a problem because, you know, if we're using, I think we're using in this case, a syntenic block size of 5. And so it could be that many of those genes will never find a syntenic placement in this map because they are on chromosomes that are too small to really be syntenic, even in the perfect case. Now, That's not as much of a problem anymore. Most genomes, in fact, pretty much all of them that we produce now, don't have these small bottom drawer scaffolds. And for example, even human has very few of these on here. It also checks whether there might be something wrong with your model specification. Maybe there's a whole genome duplication you didn't know about, by checking for over-dispersed orthogroups. These are orthogroups that hit many, many syntenic positions across the genome. In this case, since all diploidy is 1, these are both haploid genomes, haploid representations of diploid genomes. If they hit more than 8 places, Genespace says, I'm not sure what I think about these orthogroups. And so 7 orthogroups in chicken hit 8 places and 15 in humans. That's not so bad, but they do contain 220 and 470 genes, respectively. And then it condenses these hits into arrays. And it's worth noting that the human genome has more genes in more arrays. So arrays are any set of genes that are right next to each other or very close to each other in synteny and come from the same orthogroup. This is not necessarily a tandem array. We call them a syntenic array. They could be tandem or it could be just due to paralogs nearby. All right, so once it produces this information here and concatenates these, it stores this in a file that we call the comb-bed file, which I'll show in a second after we go through what the run did, and then annotates the blast files and generates dot plots for just the raw blast hits. And then it uses MC-scanx to say for each of these blast files, is there which hits are in synteny, how many syntenic blocks are there, etc. And so it does this both between genomes and within genomes because we want to be able to highlight homologs in polyploid genomes as well. And you can see there, what it's saying is that there are 15,000 genes that are syntenic between human and chicken, and they occur in 591 syntenic blocks. And there are 510 structural variants and 354 large regions. Remember, these are gene-centric estimates. So there's probably many, many more SVs in there, structural variants. But when we look at genes, there are only that many that are obvious. It then constrains the orthogroups to synteny, integrates syntenic positions across all of the genomes. So saying, all right, for every gene in the human genome, where should it exist in the chicken genome and vice versa? And then calculates syntenic block positions. That's what's going on here. And makes dot plots. And then the last thing that it does is produces these pan-gene sets that I showed in that figure earlier. I'll just flip back to it. This is a pan-gene set here, where for any particular position in the genome, it knows what all of the other genes are that are syntenic to that and orthologous to it. Okay. So it tells you that these things are stored in a fairly complex directory structure that I'll go through right now, and then we can explore what these results look like. All right, so if we go into the Genespace data folder, in R, we can't just type in command tools, so we have to use this function dir, which tells us the directory of all of this. We can also instead say system ls-l and print them just like we would in the terminal. All right, so this is now the directory structure. If you remember before, it just had the bed and the peptide files folders. That's it. It's got all these other ones in it now. And I believe we can just visualize what some of these look like right here interactively, but if not, we can always do it in the terminal as well. All right, so what we've gone through now is what Genespace does in general, and now let's go through what it actually looks like here. So it annotates all of these blast files, right? There was an issue with copying Heads up when you're copying the code out of the PDF. It's a problem. I didn't realize this. I would have made everything single line. So this is where the blast file, just all blast hits, or in this case, sorry, this is syn hits, but it shouldn't be. Let's do it for all blast hits, which should be the same name, but all blast here. And then if we read that file in, it would look like just a standard BLAST file, but I guess we're not doing that. That's fine. So we can read this data in using the function read syn hits. This is now just the syntenic BLAST hits, which is a smaller file to look at. And if we print the head of it, this is what it looks like. It's the standard BLAST columns for the query, which is denoted as genome 1, and the target genome 2, but we use genome 1 and genome 2 here because query and target are collapsed into the unique combination. So there are only 3 combinations of genomes here: human to human, chicken to chicken, and human to chicken, and not the reciprocal. And so now we have all the BLAST hits, and we can parse these in whatever way we might want. So Genespace produces by default these dot plots, which are stored here. And if we just want to look at the raw hits from one genome to another, hopefully this loads. Sweet. And this here is what the dot plots between human and chicken look like. You can see there's a lot of off diagonal, but there are these large syntenic chunks here. And Genespace, one of the big features of it is it takes this and culls all of the background noise to just the regions that are syntenic. And so if we look at those human versus chicken syntenic hits, this is what the plot looks like. Okay. And this is, we can also, just going back to the raw hits has 2 pages, one for every hit above 50, and then one for just hits within ortho groups. And you can see how much cleaner the ortho group clustering or hits are relative to any hit with a high score. And then if we just look at the syntenic hits again, each change in color here now is a change in syntenic block, and you can see why there are 500 of them, because some of these chromosomes are chopped up into tiny, tiny little pieces. Okay, so that's what the dot plots look like. You can also look at the just within genome hits to get an idea of where there's repetitive gene content. And you can see that here, anywhere off diagonal is something that's repetitive with a score of greater than 50, which is pretty low threshold, but then here with orthogroups. So this is actually more complex than many people might take the human genome for, but when we just look at synteny, it's nice and clean, just 1-to-1. Okay, so those are the dot plots. Let's go back into the tutorial here and see if I can. Might have hidden the terminal here. Let's see. That didn't do it. Hey, Damon, any idea about how to get the terminal back in here?
Through the 3 lines in the corner.
There we go.
Oh, there you go.
Yep. Grabbed it. Sorry about that. All right, so now we have this file in here, and we can do this, we can make custom dot plots using Genespace to visualize, for example, just one region of the genome. We can't visualize it interactively because we're using the R terminal here, but if you were using R Studio, you would be able to do this yourself effectively. And so we just have to paste these lines of code. This is the PDF file that we're going to write to, which is in the working directory, and it will be called custom sex chromosome dot plot. This says initiate that PDF file, which then we should see it over here. Yep, custom sex chromosome dot plot, which will be empty. We can subset the BLAST hits to anything where either chromosome 2, which is chicken, or is a Z, or chromosome 1, which is human, is an X. And then we can make the dot plot using this function ggHits, which is in Genespace, and we can add a title, for example, to it using ggplot2. And then we could change the theme to just black and white instead of the default Genespace theme. That will write that to the console, and then this will save it to the PDF. So let's open it up and see what that looks like. So now we have a dot plot here. This is the default Genespace plot, and then on the first page, and then the second one will have the customization that we did. And so you can see here the chicken Z chromosome maps to the human chromosome 9, human chromosome 5, and human chromosome 18. And pretty much there's a lot of one-to-one relationships here. There are some gaps though. Like for example, this whole chunk of the chicken Z chromosome doesn't have a orthologous synthetic genes in humans. Same goes for the human X chromosome here maps just to the chicken chromosome 4 and chicken chromosome 1. That's really highly retained. And then we changed up some themes and made the plot with a white background and grid lines instead. All right. So you can play with that. GG hits is the function that does that and lets you make those dot plots. All right, the Genespace also makes a file called it's stored in results and then the combed.txt. This contains most of the information that Genespace uses for all of its calculations. So if we open this file, this is the path to it. You can write that path differently if you'd like, but that's how I do it. And then if we load in this file and we just say, look at the head of it, it's got essentially the bed information that we had before, chromosome start and an ID, and then a bunch of other information. OFID, that's a unique OrthoFinder ID that OrthoFinder assigns. It's got the length of the peptide, the position, the genome order position of the genome here, the genome name, the array ID that we talked about before, those syntenic arrays, whether that gene is the representative of that syntenic array, the globe OG, that is that defunct orthogroup.txt classification, the globe HOG, this is the global phylogenetically hierarchical orthogroup classification. It has a flag here saying, should this gene never be used as an anchor? That comes from those orthogroups that are dispersed across the whole genome when we don't trust their syntenic positions. And then this last column here is the most important one, OG, that's the syntenic orthogroup. In this case, the globe hog qualified by whether it's syntenic or not. And so there's a lot of these global hierarchical orthogroups are split into multiple OGs. All right, so we can use and just parse this data in this combined bed file, which I'm calling CB, in a number of ways to get a lot of the information that we might want to extract out. So for example, we might want to test the hypothesis, what are the 8 orthologs, the syntenic orthologs to the HLA locus in human in chicken. So we can pull out all of the orthogroups, the syntenic orthogroups that have a gene with the HLA code in it, which orthogroups those are in, and then we can subset the whole file to any of the genes in the genome chicken that exist in those orthogroups. And we can just say, what do those look like? And so, for example, we can find that there are 3 genes that are syntenic orthologs to the human HLA. 2 of them, no surprise, are named HHLA and for the chicken. And then there's this DMA locus, which we know is also orthologous, and it's in these 3 separate orthogroups. All right, so you could also then query the BLAST hits for any of the genes that are in here. So if we just want to look at, for example, the first row in this HL bed file, this is just any genes that are within the start and end coordinates of this very first element here, just on chromosome 1. We can say, give us any of the hits that map to that position. And we can use the function query hits, if I can highlight it. Come on. There we are. And if we look at those, we can say, okay, here are all the hits. There are 7 hits that go to this position in this part of the genome, chicken chromosome 1 between these 2. And you can see what those genes are. Okay, So another thing that you might want to be able to do with the Genespace results is tabulate gene presence, absence, and copy number variation. And we have a utility function called query CNV to do this. So I'll show kind of what this looks like. So we give it the GS parameters, Genespace parameters that are just the output from the run Genespace. And if we look at this, this produces a result, excuse me, that looks like this. So for each type of orthogroup, global hogs, global OGs, those are the defunct orthogroups, and syntenic hierarchical orthogroups, we get the number of genes that are at copy number 0, the number of orthogroups that are copy number 0 in chicken, and the number that are at copy number 1. And that's what this argument, max copy number 1, is saying subsume any genes that are at a higher copy number than 1 into 1. If we set this to 2 and do this again, we'll get now up to 2 copies. And so you'll see now, for example, the human syntenic hogs, come on, human syntenic hogs at 1 copy, we're at 17,690, and now those are either present in 1 copy or in 2 copies and split out in that way. So this is just this max copy number here is to make parsing and visualization simpler for the user. Okay, so now we have this, and we could then say, okay, what's the gene presence absence variation for global hierarchical orthogroups? And we can say, okay, well, there's 2,200 gene orthogroups that are present in human and not present in chicken. There are 1,800 that are present in chicken and not present in human, and there are 1,615,362 that are present in both. And remember, this number isn't identical because there is 2 and 3 copy gene families in there. Okay, so we can also then pull out those pangene sets. Remember, those are things that look like this. And we can do that using the function query pangenes. And query pangenes takes the Genespace parameter output and a bed file, which is the coordinates. So here I'm just asking, give me all of the pangenes on chromosome X in human that have a syntenic position in human chromosome X, but in the first 200 kb of that genome. And so that produces, it says, oh, wow, there are 4 syntenic orthogroups in that position. You can see that only one of these genes has a syntenic ortholog in chicken. One of them, NKRF, that gene is not syntenic in chicken. It's found elsewhere in the genome. And then there are 2 that are present absent. They do not have a gene at all in chicken. If we do the same thing, but instead do the chicken Z chromosome, which you can just do by specifying a different bed file, bed data frame. Now we have 7 genes that are on the beginning of chromosome that have syntenic positions on chromosome Z. And interestingly, important to note that 3 of these don't have a chicken ortholog. They only have human genes. They have non-syntenic chicken orthologs, but the human gene has a syntenic position that should be on the Z chromosome because they're all coming from chromosome 18 apparently in this position. But they're absent in the chicken. So even though it's absent there, we've interpolated the syntenic position of these genes, and we should be able to pull out their IDs even if they don't exist in the genome of interest. And so you can specify this and play with it in any region of interest that you want. All right, so lastly, Genespace makes these riparian maps, right? These are plots that look like this, or this, right? Tracking regions of interest across all of the genomes phased by a particular genome. So in this case, it's just coloring the syntenic positions in all of these genomes by the chromosome, the rice chromosome. All right, so we can make these using the function plot riparian. They're also stored by default in this riparian directory here, which contains source data and riparian plots. So if we just want to look at one of them, let's just look at the chicken gene order riparian plot. That looks like this. And so you can see, remember, the Z chromosome comes from several human chromosomes, including chromosome 5 and 9. Thought it had one on 18 too. Yeah, it does. It has one little chunk on 18 as well. So you can do this. It's phased by this. In this case, it's by gene rank order, and the genomes have relatively similar number of genes, even though the chicken genome has all these chromosomes that are much smaller. But we could also do this phased by base pair instead of by gene order in this plot. Now you can see all of these little tiny chromosomes in chicken and where they go in human because they're much smaller physically, even though they have plenty of genes on them. Okay, so this is the main output. Typically, you wouldn't just show two genomes because if you do that, you can just color them by their one genome's chromosome of interest. So it's pretty trivial to do, but it becomes more computationally complex when you add more and more genomes to it. And for this demonstration, we're just doing the two genomes because it's fastest, but you could do this with literally any number of genomes. I've done it for over 100. Okay, so how to customize our riparian plots. So it's worth noting that there is a full tutorial on how to make different riparian plots here. This is linked in the README on the GitHub page. You can just click on customize your riparian plots and it will open this HTML. And so it goes through how you can make different riparian plots with different parameters. This is the raw output for 5 genomes that we use in the test case on this. And then you can customize it. In this case, you can use base pairs instead of gene order. You could, here we're reorienting and subsetting the number of genomes we're using, in this case just 4, and having different orders. You can tell it that you don't want to reorder by synteny, or you do. In this case, now all of the chromosomes are ordered by their numbers, not by their syntenic positions. So for example, here you can see human and mouse look fairly syntenic to each other here, but if you order by their chromosome names, they are all over the place. There's many other parameters that you can do. You can custom order and put the X chromosome on the left side. In this case, that's how we're doing it here with humans. You can change a variety. There's just a ton of ways that you can parameterize it. You can also change the appearance. Here we're highlighting inversions in this ugly green color. We can change the shapes of the chromosome segments to make them more square and put space, for example, between the ribbons and that. This parameterization is one that I know several people prefer. We can only label some genomes and not label others. And you can change the background and highlight regions of interest. All right, so here, using this data, we'll just go through a couple ways that we can customize our genome, our riparian plots. So here I'm making a bed file that contains, I'll show you what this looks like. So X, Z bed. That looks like this. It's just got a color for the human X chromosome and a color for the chicken Z chromosome, and that will come in later. And then since we can't do this interactively, we'll save it to a PDF, hopefully. So that's the PDF file that we'll write it to. We can initiate that PDF. It's going to be really wide and narrow because we'll only have 2 genomes. And we can see it pop up over here, custom riparians. And then we'll make 3 different riparian plots. The first one is just the default. The second is using base pairs, not order, gene order. And then the third is using gene order, but only highlighting the X and Z chromosomes. And then we can close out that PDF file and we can look at it. So here are our riparian plots for this. It's smaller. And so you can see this is gene order. And you can see the scale bar is in genes. This is in physical position. You can see the scale bar is in megabases. You can see now, wow, there's the Z chromosome in chicken is really a lot bigger than most of the autosomes. And then if we just highlight the syntenic positions between the Z and the X, you can see all of a sudden now it's really clear that one block comes from chromosome 18. All right, so that is it. That is the full tutorial that I have planned for today. There are other functionalities for Genespace, including quality control of your genomes and visualizing contigs and things like that, but we are not going to go through that today. All right, so any questions or maybe issues getting it going?
Right, there is at the minute there is one question, or a couple of questions in the Discord now, actually. Does the
Okay.
parsing filter out isoforms?
So I'm just opening up the Discord channel right now. It's going to take a second. But so isoforms, I think what you're describing there are alternative transcripts. And you can just ask on with your, face instead of fingers on Discord. But no, I'm fine to answer the Discord questions as well. So those are like alternative transcripts for the same position. And typically you don't want to think about alternative isoforms when you're thinking about orthology. And the reason for that is because you want just a single representative of a gene in that graph. If you add another isoform into it, it's possible that it will complicate the orthology graph to a point where other genes are similar to it. So you could imagine you have a nice long isoform that codes for the most common primary transcript, but then you have a little one that just is like the first exon in a piece of the last exon and has a different alternative splice. It might be that that little isoform, while 100% identical to the longer isoform in one direction and not in the other, right? Because there's a lot of sequence missing, maybe has a domain that's very similar to another gene on a different chromosome that has no real evolutionary history with that locus, but they've both co-evolved a similar domain because whatever, it's passing through the membrane or something like that. And so you can get this pollution of your orthology graphs that way. So typically you want to use the longest isoform in your analysis. The parse annotation function does that and chooses the longest isoform. Or you can do it custom and choose a primary transcript, which may be defined as something other than the longest, maybe the most common or something like that. And that's totally fine too. But I strongly discourage using multiple isoforms in the analysis initially. Now, then after the fact, if you have an isoform of interest and you want to ask, okay, what's going on with this isoform? You can build a gene tree with that too, and do that in a custom way, but I wouldn't use it in the default specifications.
Cool. Can you get microsynteny plots?
So microsynteny, I imagine you're talking about like the MC ScanX output where you're just looking at like 6 or 12 genes or something and asking, are they syntenic? Or maybe you're asking microsynteny is also sometimes defined as like really, really dispersed synteny between really anciently diverged genomes. In the first case, yeah, you can choose whatever you want to plot. You read in the hits and then you use GG hits or just any plot that you want to do that. So here I can show you quickly how we could plot microsynteny. All right, so if we go into here, we had what did we call it? Maybe AB hits. Is that what I called it? There it is. All right, so we have this these hits that are AB. If we want to just say what's this microsynteny, just in a really small scale, let's say the first part of chromosome X in human, we might say AB, right? Did I call this AB? That's what I called it, yeah. So chromosome 1 equals X, and genome 1 does not equal genome 2. So we're just asking about synteny between the two, human and chicken, not within human and chicken. And we'll just say the first 500 kb of the X chromosome. So start 1, the start of genome 1 is less than 583. All right. So, and we also have to say, and block, we have to make sure that we're not, I think all the block IDs, since this, in this case, block ID is guaranteed to have something because these are syntenic, but you have to say is.na block ID. So now this is any gene in the first 500 kb of the X chromosome. All right, so if we say, well, maybe I chose a too small of an interval. There are only 3 genes here. All right, let's do, instead, let's do 5 megabases. All right, so there are 22 in the first 5 megabases. And if we just want to plot this, say, I think it's, what's my working directory? 1 second. Okay, so we can just say, and then we can just go GG hits, MS, and then we can look at this test.pdf, and you can see it's, maybe we should do a smaller plot. Let's just remake this so that it's a little smaller. Now you've got your microsynteny plot for just this tiny position between chromosome X and chromosome 1 right here. And this is gene rank order. I guess if you wanted to do it by physical position instead, you would just add in here, use order is false. And now you'd have your physical position mapped between the two. And so that would be like a microsynteny plot. I'm not sure who asked that question, but is that what you were looking for?
Hi, I thought I'll just jump on. Yeah, that looks great. It's always a thing we want to look in at more focused regions on the genome, especially for if a gene is moved in and out of a region. So being able to look at the microsynteny in a more detailed level like that is really informative for what we're interested in.
The issue with the microsynteny, though, if one gene is moving in or out, Genespace really, that would be, if it was just one gene moving into that position on the X chromosome, Genespace wouldn't see that as a syntenic position. We set the default
Yeah.
is to have 5 genes in its syntenic block to be retained. So you can still do this. You just have to, here, I'll show you how you can do it. You have to use all of the blast hits, not just the syntenic. And so here, I'll share that, and I'll just share this and show you how it would work. All right, so so if we have something called, we'll just call it all blast, it would be read all blast, I think. And it would be syntenic hits. I think I wrote this in one of the tutorials. Let's see. Where is it? Oh, it's over here. Oh, right. I just ended up just doing this, all of the syntenic blast hits just because it's faster, but we can easily do it like this. So, but it's not hits, it's coded as all blast. All right, and then all blast looks like this. And so you can see there's a lot of things with an NA on block ID. So then if we're going to do this with test PDF, let's make it bigger because I think we're going to get multiple chromosomes here. And we want a subset. So now we're going to change this. We're going to turn off that it has to be in the same block ID, right? So it can be a non-syntenic hit there, same as before, and then we'll just call this all blast. And then we do the same plot, but let's just see what this returns. I kind of think this is going to give us something that we don't want, and we'll find out in a second. All right, so this didn't give us what we wanted. It's still just the syntenic positions, I'm pretty sure. So yeah, so it goes to all of the human chromosomes. So let's
Oh yeah.
let me figure. I wonder if I can get the help file here or not. Excellent. So the GG hits functionality. Oh wow, it's interesting. All right, I have no idea how to get out of this. That'll be interesting to resolve later.
Probably just Q.
Maybe, let's see. Yep, that did it. All right, so let's not do the help file that way. That was obnoxious. I'll just go quickly and look at what the source code is in here. All right. So it has type. So I'm pretty sure that's the one I want. Let's make sure before I do anything more. There, you would just hit question mark and then the function if you were doing it yourself. Yeah, so type is the parameter and we want all. Okay. Sweet. That didn't work. This is a problem with live coding. All right, so GG hits should take, oh, maybe color by blocks is the other way to do it. There's other ways to do dot plotting. Let's see if that did the trick. It should look terrible. Yep. So it looks really bad because we have all of these little chromosomes on it. But
Yeah.
this would be the sort of, and yeah, it's kind of garbage. The points are there. It's just that you really need a different way to do this.
Yeah. Yeah.
Yeah, I can think of a lot of ways to do it. You would want to basically just make a custom plot with all of those, or you can use that all hits and just subset those in a different way. Sorry, I didn't have a better answer for that for the non-syntenic ones.
No, that's fine. Thanks, John.
Mhm.
I have a brief question. I mean, kind of tangential to this, related to the anchor alignments. So you're using MC ScanX. I think it's depending on like 5 genes to generate an anchor.
That's what the default is, but you can set that to as low as 2 if you want.
So yeah, I guess I'm curious on parameterization of MC ScanX. How do you decide maybe thresholds of customization, and then also whether you've tried other protein alignment tools to generate anchors, and what you've seen, or if Genespace is really kind of tailored specifically to parsing MC ScanX results.
Well, all right, I answer that question, those last two really effectively. So Genespace is definitely designed to only parse MC ScanX output. And the main reason for that is because MC-ScanX can just take a custom gene-to-gene and score data set. You don't need all this other stuff. And so one thing that Genespace can do is run MC-ScanX within blocks. And some of these other software have a lot of overhead. MC-ScanX can do, 500 blocks really, really fast just by passing these small strings to it. So that's why we use MC-ScanX. The other thing is that all of these Synteny search algorithms, they all suck by themselves in positions that are complex. So if the like MC-ScanX finds literally thousands of inversions nested within syntenic blocks that are true inversions in some of these whole genome duplications, but it doesn't it encapsulates the small inversions of like 2 or 3 genes within a larger block. So it says it's a duplication when it's not. There are other programs that do a really bad job in other situations. The reason we choose Genespace is just because, Genespace chose MC-ScanX is because it was kind of the first and easiest to use. And we know where I've been working with it for a decade, and I know where its problems are, and I know how to overcome those. And so that's why Genespace does that aggregation of the array IDs and just using these array anchors. And it gets past that problem well, it doesn't get it gets past one problem that MC-ScanX, which it chains diagonals together. The other problem it gets past by not using MC-ScanX at all to determine block breakpoints. So it uses MC-ScanX to determine syntenic anchors, and then along those anchors, it uses a density-based scan to ask if there's a jump of more than 5 genes, and then it will break that block itself. So it doesn't actually rely on MC-ScanX block calls at all, and you shouldn't either. Even in the best tailored blast results, you're still going to get weird block calls. MC-ScanX is fantastic for determining hits that are in collinear hits, but not good at determining what the block breakpoints are. And then you had another question: how do you determine the what parameterization to use? So we use 5, not only because that's what MC-ScanX defaults to, but in my experience, as you go lower than that, you start finding non-real blocks more common. Like it just happens. If you use a block size of 2, there's going to be a ton of places in the genome where there are 2 genes that are in the same orthogroup that are nearby, but just by chance and are not really syntenic. And so you get a lot more noise. And if you go higher than that, if you're in genomes that are really collinear, you might get a little bit improvement in clarity. You might throw away some things that don't look real, but there's not a huge improvement.
Like above 5.
Yeah, the reason that you would increase your blocks, a minimum block size to 10 or 20 or 50 or whatever, would be if you're trying to really disentangle 2 extremely closely related whole genome duplications. So like if you just run default Genespace in maybe cotton, like one of these genomes that has very similar subgenomes, cotton's a tetraploid, you'll get a little pollution. The A subgenome might hit the D subgenome once every 1,000 genes or something like that. And so you'll get this little off-diagonal pollution. But as you increase that block size, and we know that all the genomes are super collinear, so you don't actually lose any precision when you increase the block size, but you'll reduce that noise. That's the main thing that you would want to parameterize in the run. But there are like 30 or 40 parameters that you can give in a Genespace that will let you fine tune it if you want. We just kind of don't mess with them for the most part because it's pretty robust. Yeah, the use of the orthogroups is what really makes it robust, because orthogroups are built with this graph construction framework and are not biased by, scores and partial overlapping and things like that. So it's fairly robust to any parameterization. In fact, OrthoFinder doesn't really have any parameterization that you can do to it that it makes sense. You just sort of run it at default because it's doing this graph clustering.
Yeah, we're struggling with a hex. I'm struggling with a hexaploid right now, but I saw your paper on the your decaploid, the sugar cane, which was super impressive. But did you alter GeneSpace parameters and MC-scan results to kind of adapt for that, or were you using just a five gene anchor?
This is a great question. I'm going to illustrate it for you. So if you took any two genomes that are from the same species, let's not use sugarcane because it's whale. Let's use cotton or something that has undergone a whole genome duplication. So cotton, let's use cotton because people know what it is. It has an A and a D subgenome. So if you look at any cotton genome, you'd have subgenome A and subgenome D, and they're diverged by, I don't know, 1,000,000 years or something like that. And then you'd build a tree out to, cacao or something like that, that is an outgroup. What OrthoFinder will see as typical orthogroup is anything that coalesces back here is what OrthoFinder will see as a single orthogroup. So any gene in the A or D and D subgenome that has an ortholog to cacao will have a gene tree that matches this topology. So the A and the D subgenomes will be in the same orthogroup. And so if your goal is to map genes from the A subgenome to the D subgenome, you want to ensure that you have outgroups in there because the A and the D subgenome, these will be viewed as paralogs, right? They diverged along one branch, single copy, and then were diverged into two. And so maybe you would add 2 or 3 outgroups to this run so that you would get connections between the A and the D subgenomes. Now, if the goal was to instead you no longer want to look between the A and D subgenome and for genes that are shared, but now you want to look at 2 cotton genomes that both have the A and D, and the cotton genomes diverged something like this. So you have A1 and A2 and D1 and D2, and you did the same run, but you were only interested in A versus D, you would still get all of this aggregation between A and D, which would be a problem, and it would pollute your run. So instead, what you would want to do is you would want to exclude that branch. You wouldn't want to use cacao in it, and you would just do a run that looked like this, the phylogeny, where right. So then the tree would look like that. And then any of the genes, you'll basically get a split then between the subgenomes. So you'll never get any of these deeper branches, and you'll get only trees that connect the A to the A and the D to the D. So the way to get around this problem isn't to parameterize the GeneSpace run itself. It's to change the phylogenetic scale of your to change the phylogenetic scale of your run to include an outgroup. And GeneSpace, the init GeneSpace functionality contains an argument called outgroup or ignore these genomes. It's identical. And essentially what it does is it runs OrthoFinder with those outgroups in it, but then doesn't touch them for synteny. And so if you want to compare the A and the D subgenome in cotton, you'd want to include cacao and maybe like common bean, poplar, things like that you know are going to help coalesce genes between the A and D subgenome. And does that answer your question?
Yeah,
Okay.
that's super helpful. Thank you.
Yep, no problem. And that discussion is at the very end of the methods of the GeneSpace paper that describes what situations you'd want to use outgroups and which ones you wouldn't. Damon, are there any more questions on Discord?
No, not at the minute. I had a very distantly related because this isn't my field whatsoever. This is the sort of thing I'm looking at going into with like Hymenoptera. So they're like
Nice.
haplodiplo
Yeah.
genomes. So I think could be quite interesting to have this sort of pot going for them. Is there any sort of plans of where to take Genespace in the future at all?
Thanks, Damon. That's a good question. So I think the Hymenoptera I haven't looked in that system at all. I know I've looked in Lepidoptera and a couple others. There's a really nice paper on butterfly genome evolution out there right now that looks at chromosome synteny. It's beautiful.
Yes,
I
the
imagine
Charlotte
having,
Wright's paper.
yes, that's right.
Yes.
Um, and there's, I mean, you'd imagine a haplodiploid system. You'd hope that there wouldn't be that much major structural variation because if some of your progeny need to survive at the haploid stage, you'd want to make sure you give one copy of your whole genome to that individual and not two copies of chromosome Y or 9 and 0 copies of 8 or whatever. So I would imagine that those genomes would be pretty conserved, and that could be a cool question to ask. In terms of next steps for GeneSpace, well, there isn't a lot of funding for developing software like this. So I would love to hire someone to write it in Rust or C++, preferably Rust, and have it be a command line. But right now we don't have that. We did secure funding and have built a gene-free GeneSpace called DeepSpace, which is also an R package, and can do multiple genome synteny incredibly fast, faster than GeneSpace. And basically it does it through windowed alignments. So you can do like, a whole genome alignment, but I mean, it's windowed, so it's simplified, but genome alignment of multiple like human scale genomes in like 5-10 minutes, something like that. Whereas on your local computer, instead of if you had to do like whole genome alignments using Minimap of 2 human genomes, that would, you couldn't do that on your personal computer because it fills up the cache space. And even though it's really, really great. So if you just want to make synteny maps across multiple references, that's a great way to do it. I just did 12 strawberry genomes. Strawberry is an octoploid and is 800 megs, so 1/3 size of the human genome or 1/4 the size of the genome. But it took 8 minutes or something like that on my machine. So that's the next step. Typically, what I'm trying to do in general is to make these software light and easy. And part of that is because often, like, I'm an ecologist by training and an evolutionary biologist, and often you have hypotheses that you want to test, but there's like nuance to it that you don't know going in. And if doing something once takes days, you're more likely to just do it that way and not try and fine tune and make it perfect. And if things just take minutes, you might do it again and maybe add an outgroup like Sam proposed or something like that to make the analysis more robust. And that's what we're trying to do.
Cool. So it's essentially GeneSpace as if you have protein annotations for your genome, DeepSpace if you don't.
Yeah. And
Yeah.
also I'll contextualize that deep space and whole genome alignments are really bad when there's variable ploidy. Or a recent whole genome duplication. There was this great tool that's put out by NCBI called the Comparative Genomics Viewer, I think, CGV maybe. And it works fantastic, so great in mammals and birds. And there's just no situation that it works well when you have a polyploid in there because the primary alignments and the secondary alignments have no meaning anymore because you'd imagine the primary and secondary are both primary biologically, even though that one is primary and one is secondary from an alignment perspective. And so trying to figure out how, and that's not an issue with the NCBI genomics viewer, that's an issue with the way alignments work. The reason that GeneSpace works great with whole genome duplications is because OrthoFinder lets you connect multiple nodes across one genome to a single node in another. because it's using protein, a full graph, all pairwise alignments of proteins that you could never, I mean, yeah, you could possibly do all pairwise alignments in whole genome alignments, but the amino acids evolves more slowly. It gets you more precision in the graph construction. And so basically the take home from all this is if you have any variable ploidy or a recent whole genome duplication, you really need to be very careful when you're exploring your whole genome alignments. And I would not use Deepspace for that at all. You would want to use Deepspace for single copy or things where you're comparing. Like you can easily compare two switchgrass genomes to each other because the whole genome duplication was 5 million years ago, and most switchgrass genomes coalesce in the last 1 or 2 million years. So that's easy enough to do, but you would never want to do like, I don't know, Xenopus to like the tetraploid to the diploid or something like that, and you'd get really wacky alignments out of that.
Right, so genes with hypervariable regions in deep space are a no-go then, sort of thing.
Well, yeah. So
Yeah.
it depends. I mean, so Genespace, that should be okay because a hypervariable thing, it'll just have really long branches, terminal nodes, leaves on the tree, but they'll still have the same internal structure that you would expect, hopefully, unless there's reversion back to a previous amino acid or something like that that's really messing up the tree. But in deep space, if it's really diverged, yeah, it can be a problem because the way it works is it asks for this interval minimap, where is the best place that it goes, and it pulls that alignment. And if it's really, there are many more ways to parameterize that and say, okay, if it's below the 5th quantile of alignment scores, toss it, because maybe you don't want these hypervariable alignments in there. But yeah, it's definitely, that is, I mean, I think I put the initial version 0.1 or whatever of Deepspace on GitHub like 2 or 3 months ago. So that's very much a beta release, and I'm working on optimizing it. So but it does make the pretty riparian plots the same way that Genespace does, but it doesn't do anything else really.
Cool. Thank you very much. Right. Does anyone else have any questions then?
If questions do arise, probably the best way to ask them is as an issue or a comment on the GitHub repo for Genespace. I try to stay up to date on that. I know there are a couple outstanding issues, but that's how it goes. But I'll reply to you within a week, unless it's something that pops up that I have a really quick answer to, and then I'll try and get to it right away. So yeah, please contact me there or reach out on Twitter or email or Blue Sky.
Cool. Thank you. In that case, thank you everyone for coming. Thank you, John, for giving it your time to run the session. It has been very interesting, so thank you very much. And I will see you all in another session.
Thank you very much.